It's really an honor to be here. And I'd be very happy to get your diverse perspectives, both on the experiments and the theoretical aspects of my work in the end as we talk. So what I'm going to show you today is a summary of the experiments that I've done in the past couple of years on the mechanisms of neural sequence generation underlying Birdsong, which is a complex learned sequential behavior. This is on the other side of neuroscience that I wanted to explore after I left Matthew Diamond's lab, working on the sensory and perceptual kind of questions in neuroscience, moving more towards motor neuroscience and motor control. Now, in particular, today I'll be focusing on the role of interactions between thalamus and cortex to test hypotheses of cortical sequence generation. Now, what do we mean by sequential behavior? Let's first clarify this. So motor behaviors are built out of a sequence of essentially muscle contractions that evolve through time. Now, many of the amazing behaviors that animals perform are those that are learned, and they depend on the sequential muscle movements to produce a seemingly continuous and smooth motion, whether they are ballistic behaviors like a tennis serve, or ongoing behaviors like speech, writing, typing on your keyboard, or playing the piano. But the big question that has fascinated many of us is, how does the brain orchestrate these sequential behaviors, and how does it learn to produce these sequences to begin with? Now, we all have controlled our muscles as we woke up today to tie our shoes or brush our teeth. And in fact, some of the most impressive examples of motor control are those that require extreme precision. For example, the example of the tennis serve or a choreographed dance, they require precision, but even something as simple as tying your shoe or brushing your teeth, as those of us who have little babies know, is learned very slowly with lots of practice and trial and error. And what we want to understand is what's going on in the brain when we are executing a skilled movement, and how we learn to do these movements through practice. And to understand how motor control and motor learning works, we chose to study one of nature's natural learners, the songbird. And one songbird in particular, the zebra finch. Now, an adult zebra finch sitting here on this branch sings a very precise and stereotyped song that sounds like this. Now, this here shows a spectrogram of the song. A spectrogram is just a heat map of power in frequency bands over time. So here, the x-axis is basically time, y-axis is frequency, and the color shows the power of that frequency at that time. So it's just a cool way of showing like sound in pictures. Now, as you can see from this spectrogram, the song is composed of several parts that we call syllables, and they are sung together in a repeating motif of about half a second to 1 second long, and usually they're like somewhere between 2 to 7 of these syllables strung together in a song. Now, each zebra finch sings only one song that they learn from usually their natural father. And I should say that this is a sexually dimorphic behavior because this is their courtship song. So they only male birds sing. So the juvenile bird sitting here next to his father will listen to the adult tutor in a sensory phase, and will memorize the song of the tutor, and then he begins to sing on its own. And they start out by producing a highly variable babbling called subsong that sounds like this. And slowly, over the course of about 60 days or so, their song becomes more stereotyped, and slowly some repeated song elements gradually start to emerge. So we call this a plastic song. And finally, as an adult, they sing a song that's a good copy of the tutor's or their father's song. And if you compare the imitation to the father's song, you can tell that actually he did a pretty good job. So let me play that again. All right, so studying this amazing system has led to a simple reinforcement model of song learning introduced by Doya and Sainosky in the late 80s. So let me give you a quick overview of this. So there's a part of the brain that produces the song that's initially, as you heard, a poor copy of the tutor song. Now, there has to be also some variability to allow the bird to explore different vocalizations and see what sounded best. And then the bird also has to have stored memory of the song somewhere, perhaps in his auditory system. As he produces a song, he hears himself, and he evaluates the quality of his imitation by comparing that song to the auditory memory that he stored from the tutor song. Then there has to be an error signal fed back as a result of this evaluation, whether he made a good sound or not, going back to the motor and variability generating circuits. And over the course of time, the poor match of the song gradually converges to the good match to the tutor, and the error slowly diminishes and goes to zero. So today I'll use this general framework and talk about the part of the system that's responsible for the temporal control of neural sequence generation. Or simply put, how does the songbird's brain keep track of the time in producing this behavior? And in particular, I'll tell you about the thalamocortical dynamics involved in song production. But first, I need to summarize and explain a simple model of neural sequence generation as we think is happening in the songbird brain. And then I'll test two incompatible models for the temporal control of song. So in the songbird, the song is controlled by a midbrain thalamocortical feedback loop. The interesting thing about this system is that it's a separate circuit that's exclusively dedicated to singing. So here you see a diagram of the main parts of the system delineated over the course of the past few decades. Many anatomical and lesion studies have mapped out this set of connected brain regions that are all needed to produce the adult song, which we call the descending motor pathway, as you can see in these red arrows here. Now, the premotor cortical region, HVC, here at the top, projects to a motor cortical region called RA. Names in bird brain are a bit weird for those of you who are more familiar with the mammalian system. Now, HVC and RA are thought to be analogous with the mammalian motor cortex, with RA neurons synapsing directly onto motor neurons in the brainstem, just like our own motor neurons down our spinal cord. And these motor neurons will control the vocal organ or syrinx, as well as the respiratory centers. Now, the brainstem sends a feedback to a critical part of this network, which is called nucleus Uveaformis because of its grape-shaped anatomy, which is a tiny thalamic or motor thalamic region that's absolutely necessary for singing. Basically, if you lesion this tiny nucleus, the bird loses the entire ability to sing. So Uva, in turn, projects back to cortex, to HVC. And basically, many of the areas that I talked about in the songbird brain are thought to be analogous to the brainstem-thalamic, basal ganglia, and cortex of mammals. So if you put an electrode in HVC up here, as the bird is singing, you'll see that neurons projecting to the motor pathway RA burst at most once per motif. When you align these spikes to the song motif, it becomes clear that each projection neuron fires at exactly the same time in the song every time the bird is singing, with sub-millisecond precision. So they are extremely precise. And here I can show you, and here you can see that each tiny vertical line is basically a spike, and each row in this raster plot is one rendition of the motif. So you can see that the projection neurons fire in a burst of action potentials that last about 5 to 10 milliseconds. Now, when you look at a group of these neurons, if you record from many of them, they all have this ultra-sparse activity that's locked to a given moment in the song. But different neurons burst at different times in the song. Now, these physiological observations have motivated a well-supported model of HVC function, a hypothesis that says HVC basically produces a time basis that governs song production and perhaps even song learning. So this hypothesis was inspired by two main initial physiological observations. First, that there is a gradient of temporal sparsity between HVC and the downstream regions to it, like RA, which you can see by just comparing the sparsity of the different neurons here in different colors to the recordings made in RA. And second, the fact that HVC bursts are tightly coupled to bursts of activity in RA. Basically, if you do a paired recording, as I'm showing you here, of RA and HVC neurons, you'll see that the RA bursts always follow the bursts of activity in HVC. So in this model, the population of HVC projection neurons burst continuously throughout the song, forming kind of a clock that governs song production and learning at every moment in the song. Here I have a cartoon of this model where the population of HVC projection neurons are active in sequence. And as I step through the animation, you'll see that they tile the song and they drive the pattern of activity in the motor cortex required to produce the right sound at the right moment. So let's say it's timestamp 1, and then 2, 3, 4, 5. All right, so the data that I showed you so far from HVC was only from 9 projection neurons, because recording from these neurons is extremely difficult for reasons that I'm happy to get to after the talk. But if you somehow manage to record from many more neurons and show the bursting activity as a heat map, it becomes obvious that HVC neurons burst throughout the song. And this seems to be, as I'm showing in these 5 example birds, this is a very nice neural backbone for making a clock or a timer. So I've shown you that RA seems to be encoding the vocal motor sequence as a complex sequence of bursts, like as you saw from that complex picture. And HVC seems to code for temporal order by a highly sparse sequence of bursts. But the question is, where are the dynamics? How are these sequences in HVC generated? And basically, where is the timer? Who's dictating the time? There are several models about how you can get a neural sequence like this, like the ones that I've talked about so far, and they all involve circuit models of the thalamocortical loop that I mentioned. And all of those have experimentally testable predictions. One possibility is that the dynamics are only and simply within HVC. But it's also possible that these neurons are simply driven by something else. For example, the thalamic input that we talked about. So for simplicity, let's consider two extreme alternative possibilities. One possibility is that neurons in HVC essentially form a synaptically connected chain where neurons activate one another with a latency of 5 to 10 milliseconds. So basically, first this group is activated, then the next group, and then just simply a chain of activity in cortex. And this activity basically propagates through the chain to form a continuous representation of time within the sine motif. Now, importantly, this model says that because sequential activity is generated by connectivity within HVC, then the temporal dynamics of the sine should also be chiefly controlled by the local cortical circuits within it. There's actually quite a lot of evidence, including from our own lab, that suggests that this is actually not one single continuous chain throughout the sine motif, but there seems to be that each 100 millisecond or so syllable is produced by a chain within HVC, such that the end of each chain reactivates the beginning of the next syllable chain through this thalamic feedback loop. So the idea is that each syllable here forms a kind of a behavioral subroutine that's represented by a little module in HVC through which activity propagates and basically just ballistically generates one sine syllable and then the next. Now, an alternative model suggests that instead of intrinsic dynamics inside HVC, its sequential activity is a result of a distributed and recurrent synaptic network activity propagating rapidly around a fast feedback loop from HVC to RA to the midbrain to the thalamic center Uva and back to HVC in a very fast cycling loop with the speed of roughly 20 milliseconds per cycle, meaning that about a syllable of about 100 milliseconds would have 5 of these fast iterations. So according to this model, Uvas upstream of HVC, its activity needs to be persistent, and it should be persistently active in order to generate and propagate the moment-to-moment sequence generation in HVC. So to distinguish between these models, we combined electrophysiology, manipulation of neural dynamics by focal cooling, anatomical tracing, and finally theory. So let's get to this. So we've seen that HVC neurons are extremely precise. The bursts are precise, and the spikes within these bursts are precise. And also, if you align these with respect to the sine, they are extremely precise with sub-millisecond precision. And because they, each neuron fires only once and no more per motif, they're extremely also sparse. The other interesting thing about them is that they are very reliable. So they always fire. Every time the bird sings, these neurons fire. So our model of sequence generation in HVC, whatever it is, should explain how such patterns of activity are generated. Now, according to the chain model, the simple chain model, it's the internal circuitry, as I mentioned, that mediates this process. But according to the loop model, these spikes are basically driven by the upstream thalamic nucleus. Now, the best firing pattern in Uva, this thalamic area, to derive such sparse and precise bursts is a sparse and precise firing pattern. So we should see what Uva neurons look like. So the electrophysiologist would just go and try to record. So for many years, our lab, among others, had tried to record from single units in Uva, but because it's impossible to record single units with conventional metal electrodes, no one could do it. Uva is extremely tiny. This area is basically twice the diameter of an average human hair. It's pretty deep in the brain, and it's very compact with firing rates during song that are extremely high. So what I had to do was to build a microdrive that allowed me to use glass electrodes to record single units from identified neurons through antidromic identification that project specifically to HVC and then record from them while the bird is freely moving and singing. So this picture shows a fluorescently labeled Uva HVC projecting neurons. You can also see the glass microelectrode on the slide just for reference. And with lower magnification, this is how deep this area is. All right. So here I'm showing you a raster plot of a recording from one example bird. Here, each color is the activity of one neuron, and each row in the raster plot is one rendition of the song. On the top, you see the spectrogram of the song of this particular bird. And these neurons are all identified HVC projecting neurons recorded in Uva. Everything is aligned to the onset of the song motif, so you can compare different neurons to one another. So before recording these single units, sparse and precise Uva patterns that I told you could be a potential for creating the sparse patterns that we see in HVC could have been a theoretical possibility. But in fact, just looking at single neurons by eye here, it turns out that this is definitely not the case. So as it immediately becomes possible to discern from these recordings, firing patterns in Uva are much denser than HVC as you compare, and they are much less temporally precise. But despite this variability, anyone looking at these patterns could tell that these Uva neurons contain a lot of timing information nevertheless. Now, one way you could generate precise and sparse firing patterns in HVC is if you have a lot of convergence from many different Uva neurons in HVC, so that the HVC neurons can find one particular time and precisely spike according to that. So we can ask, at least computationally, can you make a feed-forward model that can actually extract a precise firing pattern from the convergence and integration of these Uva signals onto HVC and make HVC fire precisely and sparsely? In other words, can we estimate the upper bound of HVC firing precision if moment-to-moment HVC firing is driven primarily by these thalamic neurons? So the answer to this question depends critically on convergence. So this would all work better if you had a massive convergence from all Uva neurons onto HVC neurons. Now, if we manage to record to label these single HVC projecting neurons in Uva with an anterograde fluorescent tracer, we see that single Uva neurons within HVC make these very focal axon terminals, which occupy a sphere of about like almost 100 micrometer in diameter. So we know from the tiny size of Uva and from several studies that we can estimate a total number of these HVC projecting neurons is around 700. And we know that the volume that these axons occupy is around 1 nanoliters. By knowing the overall size of HVC, you can basically estimate an upper bound somewhere between 2 to 10 Uva inputs possible per HVC neuron. You can also estimate based on some more sophisticated ways based on the ultrastructure. You can count the botons and put it in reference to the dendritic arbors of HVC cells, and basically your estimate will be somewhere around the same. So convergence is low. Now, in collaboration with Deje Jin's group at Penn, we set out to specifically ask, what is the convergence of Uva input in HVC that would enable it to fire as precisely, sparsely, and reliably as we see in HVC recordings? So we set up an optimal model that learns to fire in a particular window of 10 milliseconds, given Uva input from my recordings. And then we simulated HVC neurons based on both a biologically informed two-compartment model or a leaky integrate and fire neuron. So it turns out in this model, HVC neurons can produce precise and sparse bursts, which is surprisingly good, but only when the convergence from Uva to HVC is high enough to overcome the inconsistencies of Uva firing patterns that you saw. And our estimate is that this should be around 20 Uva inputs per HVC cell. Now, I remind you that this is it's not a biologically plausible model. This is just one way of trying to understand how can you learn these precise spikes given the inputs from Uva at best. So we're giving it the benefit of the doubt, and we just saw that this level of convergence is not supported by axon tracing data. So we're also getting very compelling data from Mike Lung's lab, where they recorded 2-photon calcium signals from hundreds of these HVC neurons for a long period of time, and their data clearly shows that HVC neurons are extremely reliable and they almost never fail. And the number that I've calculated is a reliability of 99%. Now here on the right, I'm showing you one example neuron that was recorded over 60 song renditions, and you can see that this neuron never fails to fire in its window. So basically, these overall, these results suggest that the firing patterns in Uva is not precise enough to actually explain the precision of HVC firing. So next, we took an alternative approach of just going and manipulating the circuit dynamics through temperature manipulation to test our hypothesis. Now, the basic logic of this kind of experiments is the following. If a brain circuitry involved in a particular brain area is involved in controlling the song timing, then cooling or lowering its temperature should slow the production of song, because the speed of brain processes, like many other biochemical processes, is strongly temperature dependent. Now, we can do this using a Peltier device or a solid-state thermoelectric heat pump, basically a miniature brain refrigerator, and then put it in different parts of the brain and change its temperature and see what happens to the dynamics. Now, lowering the temperature of HVC by a few degrees Celsius leads to stretching of the song, and in a nice linear way, and the slight increase in the temperature of HVC decreases or contracts the duration of song. And amazingly, this was shown to happen at every time scale within the song motif. So the idea is that cooling introduces an additional latency in each link inside of the system, inside HVC, in the local chain model. So basically, these links will stretch as a result of cooling. So signal propagation is delayed, and that results in dilation of sign. However, the loop model here predicts that cooling any other part of the loop, such as the downstream RA, would have to exactly lead to the same result as HVC cooling, because the dynamics run around the loop, whereas this is not the case in the chain model. So to test this, one should change the temperature of downstream RA. So I did exactly this experiment. I built two Peltier devices and bilaterally implanted them into RA. But because of the proximity of HVC and RA, just because of anatomy, cooling RA would inevitably cool HVC as well, and that would just confound our results. As you can see in these curves, as you cool RA, you'll see that it also has a residual effect of cooling on HVC, and that would just confound our results. So what I did is that I did these cooling experiments while maintaining the HVC temperature using a PID controller. So the result is very straightforward to interpret. As long as HVC temperature is fixed, the change in RA temperature has no significant effect on sign duration. And here I'm just showing data from multiple birds, and on average, you can see that they are all, regardless of the amount of cooling in RA, the stretch in millisecond of the duration of the sign is essentially distributed around 0. So these results prove that the dynamics are chiefly controlled by HVC, and cooling RA does not have a significant effect on the dynamics of the sign. But how do you know that cooling RA actually has a physiological effect on it? So in this raster plot up here, I'm showing you the tonic activity of a single unit recorded in RA while I'm cooling it. And you can see in this example neuron that the cooling leads to, in each row in different color, you see a different temperature. And you see as the temperature is reduced, you see the RA tonic spiking rate is just decreasing. And indeed, at the coldest temperatures, these spontaneous spikes nearly stop, telling us that the oscillations are probably not a source of song timing, because they're just like so radically changing with respect to temperature. But RA also has significant bursting activity, and that's what you can see in these more dense spots. So we wanted to see what happens to these bursting activity, not just the tonic. And in fact, it's been shown that these bursts in RA are driven by HVC, and I showed you some examples at the beginning of this talk, and they are the ones that are driving the sign. So to look at the effect of temperature on RA's bursting activity, similar to what happens during song production, I electrically stimulated HVC to evoke bursting activity inside of RA, like this, such as during song, while RA was being cooled. Then I record from RA neurons to measure the latency of this orthodromically evoked bursting of RA as a function of RA temperature. And as you can see in these plots, there's an average 2 millisecond stretch in the spike latency in the coldest condition, which is a massive effect considering the size of the brain and the speed of the processes here. So basically, the idea is we can use this data and we can actually estimate how much of the time or the dynamics of the song is controlled by the feedback loop versus inside of HVC. So we could actually put some experimental bounds onto these numbers. So we could then interpret the results of these experiments in conjunction with the behavioral results of song dilation as a result of RA cooling. So basically what we can do is to simply, if I just remind you that here, the idea is that every 100 milliseconds or so, you have one loop. And in this model, you have this fast loop that takes about 20 milliseconds per cycle. So each of these 2 millisecond latencies would cumulatively slow down the song in each cycle. So you would expect for each of these loop iterations, one, at least in the coldest temperature, 2 millisecond latency added to your song duration. So what you can do is to simply look at this data and estimate how many loops we have during the song motif. So in the stimulation experiment, at the lowest data, the RA temperature, we saw an average spike latency of about 2 milliseconds. And in the singing data, we can measure at the same temperature the maximum stretch in song duration, or motif dilation, as we call it. And then the amount of motif stretch divided by the average spike latency as a result of cooling would give you a rough idea about the number of these loops. So in order to get to these numbers, you need to have very precise control over your temperature settings. Otherwise, nothing would make sense. And our estimate is somewhere between 1 to 7 loops per motif, which is like about a one second motif, which is completely inconsistent with the fast loop model in which I showed you. An average 1 millisecond would basically need to have 50 cycles because it's a 20 millisecond per cycle. All right, so I'm almost done. And in conclusion, I hope I convinced you that motor thalamus, although necessary to maintain this clockwork propagation of neural activity in HVC during singing, cannot be driving every single moment in the song. And we think that this is more consistent with a conceptual model in which the thalamic inputs from Uva can activate sequences in HVC, in cortex, to direct the behavior. And this is similar to some recent theoretical proposals that suggest that thalamic input can precisely initiate individual elements in the context of a skilled behavioral sequence. I'm happy to discuss this more. We think in the case of the songbird, the way that this works is that through a feed-forward excitation mechanism that involves activation of the syllable chains in HVC at syllable timescale, and this is pretty similar in other species. In rodents, the observation from, for instance, the Svoboda's lab regarding the role of thalamic input to initiate motor cortex for movement initiation. So I'd like to thank all of my collaborators from the Phi lab, the Deje Jin's lab, and Mike Lang's lab. And also thank you again for inviting me. Thank you so much for listening, and I'm happy to answer questions.